Yeah. Hi everyone. So this week we'll be continuing our lectures on biologicals and biosimilars in dermatology. In previous 4 weeks we were discussing TNF alpha inhibitors in psoriasis, and that includes etanercept, adalimumab, infliximab and certolizumab pegol. So I received few comments who wanted to know what are the biologicals and what do we mean by biosimilars in dermatology. So I thought I'll take a break in between those drugs and discuss in brief what exactly are biologicals and what exactly are biosimilars, so that we may able to know what all class of drugs we are talking about. Okay, so with that we'll start this week's discussion on biologicals and biosimilars on dermatology. We'll be covering essentially their definitions, what is the difference between a biosimilar and a biological and generic drugs, how do we get all the licenses to formulate these drugs, and what all to be kept in mind while switching from a biological to a biosimilar or vice versa. Okay, so with that we'll start our presentation. Okay, so what essentially are biologicals? So in previous week lectures we had discussed about various biologicals, namely the four biologicals, and what are these class of drugs, which include adalimumab and rituximab and infliximab and all these sorts of drugs. So these biologicals are proteins or their derivatives that regulate the immune system or support tumor specific defense. Now tumor specific defense is not actually needed for us. The most important parts of this definition is that it includes proteins and their derivatives which regulate immune system. Okay, it is also known as recombinant therapeutics. Why? Therapeutics means it is used for treating disorders, and recombinant means what happens is we take a bacteria and then we introduce a genetic material of from the human genome into the genome of the bacteria, and when it gets translated to different to make proteins, the part that we had introduced from the human system also gets translated, and this part then is used to form various immunoglobulins or different forms of immunoglobulins or maybe chimeric immunoglobulins, and these are what becomes eventually biologicals. Okay, so remember many years ago when we were in our undergraduation, we were studying how these things were made. So what happens is that you just replace a portion of the bacterial or any vector genetic material and introduce a portion of human genetic material into it. And when the bacteria replicates, the protein also gets replicated, or the gene encoding for that protein also gets replicated, forming the required protein which it would have had formed if it was in a human body. And then the company collects all these formed proteins, manufacture it properly, purify it, do all sorts of post-translational modifications, and then form the biological. So essentially, biologicals are proteins or derivatives which are derived from a biological living system, a living source, and are responsible for regulation of immune system. Other things that are also known as biologicals include serum. It also includes vaccine, toxins, also blood components, and even for various gene therapies. These are all forms of things which can be called as biologicals. However, while in the present context, by biological we mean all the biological therapeutics that we use in dermatology. Clear? Now, another definition includes that these are pharmaceutical compounds which are synthesized or extracted from a biological source. Remember that in the last slide we discussed that it is derived from a living organism or translation of genetic material of that living organism, and thus it comes from a biological source. And they have highly complex structures, and because of this highly complexity they are very costly. Okay, since you are using a biological system to formulate these kind of proteins, the system essentially becomes very costly. And the structure of biologicals are so complex that nothing should go wrong in the manufacturing process, and there has to be multiple checks and balances, and the process has to be free from all the errors. and that increases the cost of these biologicals, and that is why you see that all sorts of biological molecule or the innovator molecule or the original biological molecules are very costly. So these are divided or not divided, classified into four parts: monoclonal antibodies, which are the basic area of discussion in this present lecture; receptor modulators, enzyme modulators, and even hormones. We all remember that insulin is one of these biological which was created by the company in USA, Eli Lilly, and this had revolutionized the treatment for diabetes all over the world. And because of that, now we can understand that mainly antibodies, the monoclonal antibodies, are not the only biologicals. Other biologicals also exist. However, in the present scenario, the discussion will be around monoclonal antibodies only. There are few more points which I would like to mention, which may be important for people who want to appear or prepare for quiz. Muronumab. Muronumab is a CD3 monoclonal antibody. CD3. Let me just write it again. It is a CD3 monoclonal antibody, and it was the first CD3 first monoclonal antibody to be made. It is Muronumab. MUROMONUMAB. Okay. After that, first fully human monoclonal antibodies, which we have already discussed in one of the previous videos, adalimumab. Clear? Now, first monoclonal antibody which got approved for psoriasis is Alefacept. This got approved in 2003, around about 2003. So this was first monoclonal antibody which got approved for psoriasis, Alefacept. It's a fusion protein from the same family as etanercept. And first biosimilar is CTP13, and this is a biosimilar of infliximab. Okay, so CTP13 is the first biosimilar to be created, and it was a biosimilar of infliximab. So these 4 points are good enough points for those people who like to prepare for quizzes. Now, as we have discussed that biologicals are classified into monoclonal antibodies, fusion antibody proteins, recombinant human cytokines and growth factors, and also IVIG. Okay, so these are the immune modulating biologicals. So in the last slide, we had all the biologicals, and from those lists, we had taken out biologicals which are used to modulate immunity only, and that is why all the biologicals that we'll be studying, or we have already studied beforehand, come into one of these categories. So they can either be monoclonal antibodies or fusion antibody proteins or recombinant human cytokines and growth factors or IVIG. Okay, so let's see what all these are one by one. First are the monoclonal antibodies. They are monoclonal, that means one clone. That means they arise from a single clone of cells. Now, what exactly clones? Clones are two living organisms which are identical to each other. When we say identical, that means the genetic material is completely same. So when these cells divide and form a lot of different cells, their genetic material is also copied, and from the copy of those genetic materials, we form the monoclonal antibodies. So they are essentially or theoretically similar. However, there are a lot of variability in the process, and that is why they are not exactly same, but they are highly similar to each other, and these are biologicals. These are not biosimilars. Okay, these are biologicals. Now, a good idea is to learn how these biologicals or monoclonal antibodies are named, so that whenever you will hear the name of a new biological, okay, a new monoclonal antibody, you will have some idea regarding what is the source of formation of these antibodies. For example, depending on the source, we have three parts. One is chimeric. So in chimerism, the genetic material comes from two different species, entirely two different species. So in chimeric, 30% of the genetic material comes from murine. Murine means rat or mice. Okay, so 30% genetic material comes from mice, and the rest is from human. And you will see an XI in between. A good example is rituximab. So can you see XI or infliximab? So whenever you see an XI in between, remember that it's a chimeric antibody. If it is humanized means that the organism in which the cells are dividing is not human, but the cells which are dividing inside that animal is human. So in humanized, you just have a 10% murine genetic material, while the rest 90% is from human, and it will have a zoo. Okay, so it has there is a biological by the name of ranibizumab, bevacizumab. These are VEGF inhibitors. Bevacizumab, usually used in diabetic retinopathy or retinopathy of prematurity. So these all are the drugs which have zoo in between. Okay, trastuzumab. Okay, remember. Now, human are that only human components are used. Genetic material is not shared by other species. It will have a U in between. Classic example is adalimumab. Adalimumab. So it has a U in between. That is because it is fully human. Now, depending on the target, we can either have immune target or interleukin target. So immune target will have a L in between, while interleukin target will have a KI in between. So these are older nomenclature. This one is a new nomenclature. In new nomenclature, we don't have KI, we just have K, and we just have L. A good example is ustekinumab. So this is an interleukin 23/12 inhibitor. Ustekinumab. So we have a KI in between. That means it is against the interleukins. Another example could be anakinra against interleukin 1. I'll just write just to complete the list. Anakinra. And if there are additional moieties attached to the parent molecule, for example, in the last last week we were discussing about sertolizumab. So it is again a zoo. Sertolizumab pegol, in which a polyethylene glycol moiety was attached. So another word is usually attached to it. Sertolizumab pegol. Clear? Let's move forward. Now, fusion antibody proteins are chimeric. Again, chimeric. So, remember what we said about chimerism? Chimerism is that the genetic material does not come from a single organism. It is a mixture of two or more than two organisms. So, chimeric antibodies are a fusion of two or more genes, which after translation will lead to a fusion protein. Okay. So, let's just recapitulate again. Let's say this is the murine genetic material or genetic material from the rat, and this is a human genetic material. When this gets translated, you will have a human part of antibody and a rat or a mice part of antibody. Now this is a chimeric antibody. Okay, but what happens when you fuse two different genes together, which after translation lead to two different molecules, two different protein molecules, and then after translation you fuse them together to form a chimeric molecule. And a classic example of this is etanercept. So etanercept is a classic example. Remember, the etanercept is a Fc combined with the TNF alpha receptor. So two different proteins are combined together to form a new monoclonal antibody, and that is why it becomes a fusion antibody protein. Clear? If this part is not clear, see this particular slide again so that you know the difference between what is a fusion antibody, monoclonal antibody, and what are fusion antibody proteins. So in fusion monoclonal antibody, the entire monoclonal antibody is exactly translated, but it is derived from two different genetic material. In fusion antibody proteins, the two different proteins are translated separately and then joined together to form a new chimeric protein. Clear? Now the names for these molecules will have a sept in between. That is why Eterna-sept or Alefa-sept. Clear? There is one more example which I am just writing to complete the list. This is known as Denileukin detox. Okay. So remember that Ki means it is against an interleukin, and detox. Dip comes from diphtheria. Diphtheria. Tox comes from toxin. So it's a recombination of immunoglobulin with the toxin from diphtheria, and it acts against interleukin 2. D usually is used for 2. Do, die. Okay. So interleukin 2 inhibitor, which is formed by combining the diphtheria toxin with immunoglobulin. So that is another example of chimeric antibody proteins. Now, recombinant human cytokines and growth factors are small immunoglobulin proteins and glycoproteins. They are released transiently after any immune stimulus, and they act through receptors to modulate humoral and cell-mediated immunity. So they are just immune regulators, and they are released whenever the immune process is going on. A lot of chemicals are released transiently so that they can have their effect, and then after the effect they are neutralized. So these include, for example, recombinant GM-CSF, which is granulocyte monocyte colony stimulating factor, or other interferon therapies. These are not much used in dermatology, so we'll skip this part now. Now, IVIG is something which is very commonly used in dermatology. These are fractionated blood products. So basically, these are artificially created immunoglobulins. So since immunoglobulins are a part of our blood, it is fractionated blood product. IVIG contains about more than 95% of IgG antibodies, and most of the time these antibodies are against another antibodies. For example, let's say in a patient of pemphigus, you have anti-desmoglein antibodies running around the serum, and if they are running without any kind of hindrance or any kind of stoppage, this will lead to disease normally, clearly. But when we give IVIG, this IVIG is going to attach itself to these antibodies and then remove the whole complex from the body. It exits out. Clear? And because of that, the disease is sequestered or stopped very quickly. So it has been approved for GVHD and Kawasaki syndrome in dermatology, not for other disorders as of now. And it has been easily used in dermatology in connective tissue disorders, bullous disorders. I've already given you example of pemphigus, and we have routinely used in our institute where I was studying for bullous pemphigoid also, and SLE we have used. SGSTN is another indication. Pyoderma gangrenosum is another indication. These are IVIG. Now let's come to the crux of the lecture, which is biosimilar. So in previous few slides, we had discussed what is biological. Biological are just protein molecules which regulate immune mediated action. Okay. Now biosimilar are, in this photo we can see that maybe there are two different entities which are manufactured totally differently from each other, but they are just similar enough to do the job. Okay. So I apologize to individual fans of these actors. I'm not saying one actor is better than the other, although it is Hrithik Roshan, but there is not much difference between two entities, so they can be easily used in indications. So even if these two entities look exactly the same, they have to have an exact same structure, and the companies manufacturing that biosimilar need to prove that their biosimilar is exactly same to a lesser, let's say to maximum extent. It should be as same so that it can get approval for usages in different disorders. So let me just say it again. If a product is a biological, so it is a patented product. That means if a company has made a biological, the recipe to make it, the protein structures, amino acid sequence, everything is under a patent. So other companies cannot make that, or otherwise they will have a lawsuit. Now what happens is the other company says that I will make a molecule which looks exactly or near exact same of your, and then we can ask the FDA to approve my molecule because it looks exactly same. So if you are doing it properly and my molecule looks exactly like your molecule, I should also get approval. And with that, we kind of bypasses some regulations, some strategies, so that the biosimilars are cheaper and easily available and faster available to the patients. So remember, two things may look exactly same, but they may not be 100% same. So biosimilar is a biological medicinal product that contains a version of the active substance of an already authorized original biological product. That means you have a product which was manufactured by a company, and this has already been authorized to be used in certain diseases, and what we do is create another copy which looks exactly the same and then ask to it to be authorized also. Okay. So let's say, for example, you have a T-shirt which comes from, let's say, Nike. Okay, so you have a T-shirt and then you have a label of Nike. If I remove that label, the T-shirt still remains a T-shirt and it will do the job. Okay, it will just not have that brand. So in that sense, if I can prove that my molecule looks exactly the same as your molecule and your molecule has received the authorization, then my molecule also works in the same way. So let's give the authorization. Now remember that in the authorization for biosimilars, you don't have to do studies to prove that it is efficacious. That study, the efficacy study, has already been done for the original molecule. I just have to show you that my molecule is nearly the same or exactly the same as your original molecule, and I need not test it on patient. All I have to prove that it is clinically as efficacious as your molecule. Okay. So biosimilars are also known as similar biotherapeutic product or subsequent entry biologic or biocomparables. Okay. So if we look closely at the definitions of biosimilar, the European Medicine Agency, so we'll keep on hearing this word EMA. In European Medicine Agency, it says that a biosimilar is not an exact copy but a close enough copy so that it shows that the mechanism of action or the safety profile or pharmacokinetics and pharmacodynamics is as same as the original molecule. Okay. Now here we see some differences between generic medication. Now generic medication is an exact copy of the medication. For example, if you have generic medicine A and then you have a brand name, let's say AMA. Okay, let's say AMA is a brand name and generic medication is just A. A and AMA are exactly the same molecule, exactly the same safety profile. The only difference is that AMA is manufactured by a big company and it has a good brand value, nothing else. While biologicals are nearly same looking, let's say it's capital A and small a, they have the same function. And if you replace capital A with small a, nothing much is going to happen. So wherever you use a capital A, you can easily use a small a. So you understand the difference between biosimilar and generic. Generics are exactly the same. Generics are not a biological component, and you have to have a safety profile and efficacy profile for generics also. While biosimilars are just similar, not exactly copy, they are also biological products, and you just have to prove that they are structurally very close to the same originator molecule. Clear? So as I said, biosimilars is not supposed to be identical, but same. It should do the job. Okay. So one reference that I found was that it can be only maximum 15% different from the original molecule. So original molecule is also known as innovator, and biosimilar should be round about not more than 15% different from the innovator. Clear? Now, biosimilar is similar to biological in the sense of quality characteristic. That means quality should be same as the innovator. Biological activity same as the innovator. Safety and efficacy same as the innovator. How it is different from a biological. Now, biosimilar can have a different structure. Remember that these are biological products. So, when a gene gets translated to a protein, a lot of things can go differently in this process. And after translation, we have post-translational modification, which includes, let's say, lipidization or glycolization or some modification of structures. Many chaperone proteins are changing the structure. So a protein can undergo post-translational modifications, and thus the structure can easily be different, but otherwise the protein is exactly the same, does exactly the same job, same efficacy, same characteristics. The structure is just a bit different. So a biosimilar can have a slightly different structure than biological. The function can have have some amount of variability. Remember that variability, a ceiling of 15% has already been kept. Function can be slightly different. Production methods have to be different. Remember that you have an innovator biological here, and the recipe to create this biological is secured by patent. So you cannot use the same recipe. You have to change something or the other. And the cost is highly different. Biologicals are costly, biosimilars are cheap. Okay. So just to recap that biologicals are biologic drugs which are produced from living organisms. Let me just change this pen. Biologicals. Now, biological drugs which are produced from living organisms and they contain components of living organisms. Clear? While biosimilars are biopharmaceutical drugs which have an active property similar to the one that has previously received license. Okay. Biologicals are medicines made from living cells, while biosimilars is a type of biological which is somewhat similar to the original molecule. Both of them have highly complex structures, but remember that biologicals have to prove their efficacy in strict controlled situations. That means using RCTs, placebo-controlled RCTs, comparison with the gold standard treatment. So they have to undergo very strict regulation or very strict process to get approved. While biosimilars bypasses all these processes if they can just show that they are to some extent exactly the same molecule as biologicals. Clear? Now this comparison is between biosimilars. Let me just change it again. Comparison between biosimilars and generics. So I'm not going to explain each and every point in this particular table. But if you want to just read it, pause this and read it. The paper from which I've taken this table, the citation and the DOI number will be mentioned in the description of the video. So you can just go and read the original paper. Now similarly, there are multiple steps involved from in vitro to pharmacovigilance in formulation of a biosimilar. So in vitro analysis includes all the molecular characterizations. Preclinical testings are in vitro studies like bioassays, receptor binding. So in this you find the structure. In this you find whether the structure is doing the job. Then you start clinical testing, that means testing on patients. So this is somewhat same like any drug approval process. Phase 1 is in healthy volunteers, in which we just find the pharmacokinetics and pharmacodynamics and safety of the biosimilar. Phase 2 trial is not required. In phase 2 trial, we find the dose required to treat the medication. We don't have to do it for a biosimilar because doses have been decided when the biological was receiving the approval. So when the biological was receiving the approval, the doses have been formulated, the route of administration has been formulated, the regimen has been formulated. We just have to replace biological with a biosimilar. So phase 2 is not required. Now phase 3 is just to show somewhat of a non-inferiority trial. However, in biosimilars we don't use the word inferior or superior. We just use similar. Yeah, we just use similar or we use comparable. So traditionally we don't use the word inferiority, non-inferiority trial or superiority trial. Okay. And pharmacovigilance is when the biosimilar has received authorization. After that you go in the market and look for any additional side effects that you may have missed. Clear? Now in preclinical analytical assessment, which is necessary for all biosimilars, you determine how similar it is to the originator molecule. And there is a component which is known as biosimilar index. Now, biosimilar index has been given by International Psoriasis Council, and this index is used to identify how similar is the biosimilar to the original molecule. So it has a lot of variables, and these variables are known as critical quality attributes. Now, critical quality attributes, these are standardized molecule. For example, let's say you score the biosimilar on these attributes. For example, what is the function of that biosimilar, how tightly it binds to the receptor, how effectively it is able to decrease the concentration of certain drugs. So all of these parameters are scored, and then you aggregate those score, you add those scores, and you give a final score to a biosimilar. And if that score is appropriate, you give licensing to the, or you proceed further for the biosimilar. Now for biosimilar, how do you name them? So remember that in previous slides we had discussed how we name biologicals, but for biosimilar, no such process has yet been established. But there is a mention that the biosimilar should have some difference as compared to a biological, so that the person who buys it or uses it knows that they are using a biosimilar, not a biological. Now, some of the suggestions are from the international non. There are as now somewhat three. Just a second. I just went out of it. Yeah. So there are three suggestions as of now. You have the international non-proprietary name INN, or you add some Greek letter or some Greek letters to the name. For example, you can call it as infliximab gamma, for example. Just an example. Or you can use a biological qualifier which is approved by WHO, and it is a 4-digit code. For example, if you read more about biosimilars, you will find for adalimumab ABBB or ABBA. So these are all biological qualifiers which are added to the original name so as to differentiate biosimilars from biologicals. Clear? Let's move forward. Now there comes the concept of microheterogeneity. Heterogeneity means that the products are different, they are not same, and micro means they are only different by a small extent. Now remember that biologicals and biosimilars are manufactured through a biological process, and this manufacturing process is itself highly variable. So all those genetic translations, all those genetic formulations, whenever the cell will divide, there will be some amount of mutations into it, and this leads to variability in the molecules, and this variability is known as microheterogeneity. However, no matter what are the variables, the amino acid sequence is expected to be the same. Okay, and if there is any difference, it should be explained that why this difference in the manufacturer's opinion, why this difference is there in biological and biosimilar, and that difference should have a negligible impact on clinical efficacy and safety. That means even if it is not exactly same, there are some differences between biosimilar and biological. That difference should have no impact on clinical efficacy and safety profile. Clear? Now the next concept is interchangeability. It means that biological and biosimilar are so same. They are so similar. They are nearly copies of each other, that they can be interchanged indefinitely during chronic course with no adverse events. Now remember that a lot of biologicals have to be used indefinitely or for a longer duration of time till the disease is controlled. And if you have a relapse, you start that again. Interchangeability means that biosimilar and biological are so same that let's say you can start with one biological, and in between you shift to the biosimilar, and then again shift to biological depending on availability and affordability. And this changeability can happen n number of times, multiple times, and those interchangeability should have no side effects. So that is known as interchangeability of biosimilars and biologicals. However, in the previous slide we discussed that there are some variabilities in the structures of biosimilars and in fact in structure of biologicals also. And since they are not exactly 100% same, these different structures can trigger certain immune responses in the body. Now remember that these are protein molecules, these are biological molecules, and these are not part of your human body. They are manufactured outside and then injected into the patient. So the patient's immune responses can form autoantibodies to these monoclonal antibodies, and thus the efficacy is decreased. A classic example is seen in infliximab, in which you have autoantibodies towards infliximab, and because of that, in subsequent exposure to infliximab, you can have decreased efficacy. So let's say you start infliximab and then you convert to a biosimilar and then again to infliximab. So because a lot of different structured proteins are exposed to the body, it can lead to a lot of autoantibodies, and these autoantibodies will eventually decrease the efficacy of that biosimilar or biological. Because of that, the ACR, American College of Rheumatism, and EULA, European League Against Rheumatism, does not recommend that biosimilars and biologicals should be interchanged. They don't recommend. Another concept is substitution. Now, substitution means the pharmacist can change the drug. For example, let's say you have an original biological by the name of A, and then you have a biosimilar which is small a. They have been proven to be effective, and they are such a good copy. The small a is such a good copy of capital A that you can use them interchangeably. So let's say I write in a patient's prescription capital A. The pharmacist can easily substitute it with a small a because he knows that these are interchangeable. So you can interchange them. So that property of biological biosimilar known as substitution. Now remember that these are the properties. That does not mean that biosimilar and biologicals have these as an inherent characteristic. You cannot change biological with biosimilar or one biosimilar with another biosimilar. It is just the property that they should have. An ideal biosimilar should be so good a copy of biological that they should be entirely interchangeable, and they can be easily substituted by the prescribing pharmacist. They do not require a change in prescription. For example, let's say I wrote capital A to be given to a patient. Patient goes to the pharmacy and it is not available, but the pharmacist says that small a is available. If the biosimilar can be substituted, the pharmacist can give a small a to the patient. If it cannot be substituted, the patient has to come back to me, discuss again, and I change from capital A to small a, and then the pharmacist gives small a. Okay. So it depends that how good a copy of the biological is that biosimilar, so that you can substitute. Another is switch. Switch just means that from biological to biosimilar or from biosimilar to biological, you can make switch from one or other. It is essentially a byproduct of interchangeability. So if biological and biosimilar interchangeable, you can switch between. Clear? The third concept is immunogenicity. So in the last slide, we discussed that a good biosimilar should be interchangeable with the biological indefinitely times with no adverse event. But in a slide previous to that, we learned that there can be inherent differences between different protein molecules. And because of these differences, you can have autoantibodies, and thus it can decrease the efficacy. Now, this formation of autoantibodies against monoclonal antibodies is a property known as immunogenicity. How immunogenic is the biosimilar? Okay, and it depends on various factors which can be from the patient or the disease specific or product specific factors. And most of the time, it is the product specific factors. That means the product manufactured is not an exact copy, and that is why it is leading to autoantibodies, and that is why it has an high immunogenicity. So most of the time, it is structural impurities or different structure while manufacture, or addition of various contaminants in the product, or maybe in the syringes which are supplied to auto inject. Remember that previous biological discussion like adalimumab, etanercept and certolizumab have pre-filled syringes, and these syringes can have molecules or contaminants in them, and this can also lead to autoantibodies. So whenever you use a syringe from the same manufacturer, you can have decrease in response because of that. Okay. So data of more than 1 year is required. So what do we mean by that? We mean that before giving approval to a biosimilar, we should have to say that even after using 1 year, the amount of autoantibodies do not decrease the efficacy of the biosimilar to a clinically positive extent. Okay, if we prove that, so we can have better chances of our biosimilar getting approved. Now the fourth point is extrapolation. Extrapolation means that data from one study can be used for approval for another disease. For example, let me use an example to explain to you. You have a biological by the name of A. A has been approved to be used for psoriasis and hidradenitis suppurativa HS. So it received the approval for the use in psoriasis first, and now I have a biosimilar small a which I got it approved from psoriasis. Okay. Now, if the biological gets approved for HS, my biosimilar cannot get approval for HS. For example, when we change the disease, like in this example, we're changing from psoriasis to HS, you always need a new license. However, the process for getting new license for biosimilar is a lot easier than getting it for a biological. Okay, so if psoriasis has received the authorization for using capital A, I can make a biosimilar small a and get approval for psoriasis. After this approval, if A also gets approval for HS, I need to file a separate petition for licensing in HS. However, let's say capital A has received the authorization in psoriasis and HS, and now I make a biological after it has received the two licenses. I can easily receive my licenses for these two diseases together. So let me just tell you again. If biological has received licenses for two different diseases and then I make a biosimilar, the biosimilar also gets the license for two diseases. If a biological has got a license for one disease and I make a biosimilar, it gets license for that disease. And after getting license for that disease, if the original biological gets a license for another disease, I have to apply again for the biosimilar. Okay, but this application process is a lot easier, and FDA just wants you to conduct two placebo controlled RCTs minimum. If the data is supportive of your biosimilar in these two RCTs, you can get approval for a new indication. Clear? If any point anywhere in this presentation or any other presentation you're not able to understand, just comment below this video and I'll get back to you and explain it again. Now, what are biomimics? Now, biomimics means that in countries where the regulations are not that strict, that your biosimilar has to be near exact copy of biological, you can get away with creating molecules which are, let's say, 80% same or 70% same. Okay, and you give them approval to be used in that country. So these violate the 15% limit that we had initially set for a biosimilar, and because of that, these are known as biomimics. For example, you had a biomimic known as kikuzumab, and kikuzumab is a biomimic of rituximab. But we had multiple adverse effects with kikuzumab, and that's where kikuzumab was taken out of the market. It is not available now. So biomimics are not an exact copy of biologicals. They are just enough copy which get approval in countries where the regulation system is not that strict. Okay. They are also known as biocopies or intended copies. That means they are not exact copies, but they intend to be a copy, or non-regulated biologicals. These are other names for biomimics. So biomimics are not that efficacious, while biosimilars are. Now these are some of the biosimilars which have been approved for use in psoriasis. So since I decided to include this slide because we had discussed previously all the biologicals used in psoriasis as of now, TNF-alpha inhibitors. So certolizumab and ustekinumab doesn't have any biosimilar as of now. Etanercept has RLZ. Infliximab and adalimumab have these biosimilars by these names. Clear? So this is just a list. Pause it. There's no need to remember it. Nobody is going to ask. And these are another list of biosimilars which have been approved for the use in India. So, for example, many of us have used Exemptia for adalimumab, or Reditux for rituximab, or Reliance manufactures Rituxirel, okay, Maptas by the Intas pharmacy. So these are all biosimilars of different biologicals, okay, and these are all approved for use in India. Okay, so this list is for India. The last list was just a general approval by the FDA. Now there are multiple problem areas in biosimilar products, and I'm not going to explain this slide because they are not much used. A good idea would be just to go through them, just read it up so that if any question comes from biologicals and biosimilars, you can easily write half a page using this material. Okay, so it includes that batch to batch should be consistent. There should be good demonstration of quality and quality assurance, and extrapolation of data to other indications. These are all problem areas. So you can just read through it, and then you can easily write few more points if any question comes from biologicals and biosimilars. Now these are three good papers that I found while researching biosimilars and biologicals. One is biosimilars in dermatology, other is therapeutic potential of biosimilars in dermatology, and third one is biosimilars in dermatology. I will I will put links to access these articles in the description of this video, so you can go click and then read these papers in detail, so you'll have a better understanding of what are biologicals or biosimilars. So with that, we end the current discussion on biologicals and biosimilars. Please watch the separate videos which I had made on adalimumab, infliximab, and other TNF alpha inhibitors. Subsequently, we'll be continuing a series on biologicals. We'll start with ustekinumab next week, which is interleukin 12 and 23 inhibitor. Now remember that you need to know the difference between a biosimilar and a biological, and also different characteristics which are changed when we shift from biological to biosimilar. Many a times we have been using biosimilars, they work perfectly in one patient, they do not work that much in another patient. So because of that, a prescribing dermatologist should have an idea about what are biologicals and what are biosimilars, and when to use which drug. So it's a better idea to go through this process, and a lot of viva questions are asked, which I've tried to cover. So if you have any other questions which have been asked to you and I have not covered them, mention them in the comment box, or you can just directly email me here, or just comment it below the video. I'll come back to you and explain it, and maybe together we can find your answer. Okay. So with that, I'll end this presentation. Go back and see other presentation on various other biologicals. The video on pathomechanism of psoriasis is good. Go through that. And if you have any other suggestions, just write in the comment box and I'll go back and see if I can prepare a good PPT on that. With that, bye bye, ciao. Take care of your skin in this summer. Wear sunscreen, use umbrella.